fungus. So the Oregon spotted frogs are true frogs in the family Ranidae. They are medium-sized, about 44 to 107 millimeters of the snout vent length. The females are larger than males. They are highly aquatic, spending almost their whole life cycle in the water. They have fully webbed feet and their eyes are dorsally positioned. And those two features differentiate them between some of the overlapping species. The similar species are the northern red-legged frog, Columbia spotted frog, and Cascades frog. There is no documented overlap with the Columbia spotted frog, but the northern red-legged and the Cascades frog have reduced foot webbing, and their ventral coloration is either green, yellow, or cream mottled in coloration, whereas the Oregon spotted frogs are more of a brick red to brown color. And I do have a picture of their ventral area later on in the presentation. They have a weak vocalization by the males, and it sounds more like a rapid tapping sound. These frogs are native only to the Pacific Northwest. They've once been distributed from British Columbia all the way to Northern California, and throughout the range, they're undergoing dramatic population declines. I'm going to focus this talk just on the Washington populations, where the star is on the big picture and the smaller map. They were listed as state endangered in Washington in 1997, and they were federally listed as threatened in the United States in 2014. So because of this, the situ program stopped when the U.S. Fish and Wildlife took over the program, and they are in the reevaluation phase to see if they are going to proceed and continue to do head starting programs. So 2015 was the last year that any of the rearing partners did any of the Head Start programs. Main causes of decline are habitat degradation, habitat fragmentation, invasive species such as reed canary grass and bullfrogs, and they do have predators at all life stages such as fish, otters, and sandhill cranes. The 6 Washington River drainages that they're found in are the Sumas River, the Black Slough, Samish River, Black River, Trout Lake Creek, Outlet Creek. And then in addition, in 2016, 13 new populations were found in the Samish, Nooksack, and Sumas watersheds. Even with these additional populations being found, they inhabit less than 90% of the historic distribution. And in addition, on this map, I just want to point out this orange circle is the site of the translocation project. And that is at JBLM, Joint Lewis Base McChord Air Force Base at either Dahlman Lake or Muck Creek. The habitat is often naturally flooded meadows that have temporary shallow water that's exposed to the sun and has submerged vegetation structures. Those eggs will be laid just below the water surface, and they are communal nesters, so they're particularly vulnerable to stranding if that water dries up, but having the larger egg masses retains more heat for the developing embryos. And you can see the lower picture, there are many eggs that represent several different egg masses from several different females. They are monogamous for the breeding season, so Fish and Wildlife is able to estimate that each egg mass represents 1 pair of frogs for their population estimating. The picture on the top is one of the sites that I collected some of the wild frogs for my master's work, and that is the Trout Lake site. It's at the base of Mount Adams, and these frogs are breeding in the coldest weeks of the winter. So at this site, I did need to break through ice to access the breeding ponds, which just blows my mind a little bit that these frogs are out there in such frigid water breeding. And this area is one of the larger wetland areas, and it is also a sandhill crane breeding area. So this was also the hardest area that I had to catch frogs in. They were the most wary. The Oregon Spotted Frog Working Group was established in 2008, and this group was established to guide the recovery plan. so to coordinate and advise on recovery activities. This group is managed and overseen by Washington Department of Fish and Wildlife and includes government, non-government organizations, and private agencies. And one of the outcomes of this group was the pilot translocation project that all rearing partners have participated in. The recovery plan provides strategies to increase population, find additional populations, and establish new populations across the geographic range. And this includes habitat management strategies to produce these results. So some of the background for the XC2 program: egg masses are collected from the wild, reared to subadults, and then released at Dalman Lake or Muck Creek at Joint Lewis Base McChord Air Force Base. And referencing back to that original map, the blue triangle is the site of the egg collection, and the orange dot is the site of the release. All institutions had similar setups. This photo is an example of the frog yard at Woodland Park Zoo. We used 300-gallon stock tanks with custom made lids with weather stripping and straps to hold them closed to prevent any predators from getting into the tanks and to prevent any frogs from escaping. The yards are gravel so that if a frog does happen to escape, it's a little bit easier for us to find it. We had two different pumps, and we had a water reservoir. So we had a pump to pump water out of the tanks to do water changes. Then we had another pump that supplied water from the reservoir tanks. All of the tanks were heated, and including the reservoir tank. So we had aged tap water that was used to fill each of the tanks, and it was allowed to sit for 24 hours to off gas and to come up to temperature. So there was no shock to the frogs by using water that was not at the same temperature. Inside the tanks, we had floating foam mats with artificial foliage, such as the picture there for the frogs. And as tadpoles, they were reared in floating net pens when they were small and then released into the stock tanks when they were larger. Each of the tanks had an air stone that provided a slow, gentle break in the surface to allow oxygenation of the water. Water changes were done based on either water quality or waste products that were visually seen in the water. And this changed depending on the frog size, time of year, and the temperature. For example, when the tadpoles were very small, water changes were done infrequently because there was a large volume of water and very little waste that was put into it. As the frogs got larger and closer to the release date, water changes were done more frequently since the temperatures were in general a little bit higher in the environment. And also the frogs were larger, eating more, and thus producing more waste. In managed care, diets provided were crickets, waxworms, mealworms, earthworms, bean beetles. In addition, since these are outdoor pens, any natural insects or detritus that falls in. Tadpoles are herbivorous, so any tree pollen or leafy detritus that would fall in, they would eat as well. In the wild, the frogs are opportunistic predators, mostly of insects such as leaf or diving beetles, and they do occasionally eat spiders. So before any of the frogs are released, all of the rearing institutions follow pre-release protocols. Each frog is measured and weighed. PCR testing is done for amphibian chytrid fungus, and a cool down period is done for several days prior to release. Initially, the heaters are turned down, and then just a couple of days before the release, the heaters are turned off completely. And the chytrid testing is important for each year that the frogs were held. Each of the rearing partners did perform chytrid testing prior to release, and we only had one group of frogs at the Woodland Park Zoo test positive for chytrid fungus, and that was the year that I did my master's research. So I'm going to talk a little bit about that now, and this is what I did. I hypothesized that plants that were reared with plants would have different bacterial communities than frogs reared without plants, thinking that there are going to be natural communities of bacteria that live in the wild that would be on the plants that would then be transferred to the frogs, giving the frogs reared with the plants bacterial communities that would be more similar to frogs reared in the wild. So I was able to collect plants from the wetlands with approval from Washington Department of Fish and Wildlife. They gave approval for the amount and the types of plants that I was able to use. And we, in addition, used plastic plants so that there was enough cover for the frogs. With our constant water changes, the live plants did not survive long. So I did have a reservoir just with plants in it and no amphibians that was used to replenish the plants in the different tanks as needed. So this was the hypothesis because this was the only change out of any of the rearing institutions that this is potentially how the chytrid fungus got into the tanks the year that this project was done. So the sample collection that I used to test for the bacteria was very similar to chytrid testing. However, it also including rinsing each frog with sterile water prior to swabbing the frog. So we were getting samples of the bacteria that actually lived on the frog and not getting any transient bacteria that might just be on the surface. I also collected morphometric measurements on each frog, and then the samples were all sent to the University of Colorado for DNA extraction, amplification, purification, and sequencing. All of the data analysis was performed in our studio, and the folks at the University of Colorado were gracious enough to host me and allow me to follow along as they were doing the data analysis and the computer programming to do all of that. This was looking specifically at the 16S region of the RNA region of the samples, and only bacteria have this, so it was easy to eliminate all other plant or vertebrate bacteria that was found, or samples that were found, not bacteria, sorry. So for this project, I did find that there were significant differences between all groups of frogs. And the results that were significant to the managed husbandry programs are that the managed care and wild populations and the pre and post itraconazole. Oh, and I think I forgot to mention that that year we did do itraconazole treatments prior to release, and I will get more into that, but I was able to collect samples from the before and after the itraconazole treatments, as well as at Cedar Creek Correctional Center, which was one of the rearing partners as well. So results significant to managed husbandry programs, the managed care and wild, and the pre and post itraconazole, all measures of diversity indicate that captive frogs had significantly higher diversity than on wild frogs. And there was a study in 2014 that suggested that amphibian skin is colonized by microbes that are low in abundance in the environment. So with the frequent water changes that we did, this would imply that microbes that are low in abundance in the environment, the frogs might not have had adequate exposure to the microbes that they would necessarily have in the wild if they're low in abundance in the environment. So we did see the differences that we expected, but not in the way that was expected. So moving on from that, some of the challenges that we had, again, the chytrid fungus, which was isolated to the 1 year that I did my master's research. And the next slide, I'll talk about what we did for treating them. The species was listed as a threatened species by the US Fish and Wildlife in 2014. So this added another level of red tape, essentially. It complicated me getting permits to collect the samples on the wild frogs that I needed for my study, and it also put a halt to the captive head starting programs. We had a couple of instances of death by Jamaican cricket. And these crickets are not normally the crickets that were fed for head starting programs in the United States or in zoological institutions in the United States. But in 2010, there was a parvovirus that that affected the domestic house crickets, and it essentially wiped out all populations of house crickets at the cricket breeding facilities in the United States. So they needed to switch the species that they were working with, and that was to the Jamaican, black-field, and banded crickets. So unfortunately, we did have a couple of these instances happen. And I think what happened was the cricket latched onto the frog and the frog latched onto the cricket, and neither of them let go. So both of them ended up dying. And then of course, cannibalism. Most frogs will eat anything that fits in their mouth, including smaller size frogs. So the chytrid fungus outbreak that we had, chytrid fungus has been found in the wetlands that the Oregon spotted frog inhabits, and there is evidence that the frogs have resistance to the disease. In an experiment, some frogs were inoculated experimentally and did not die or exhibit any symptoms of the disease. However, we did not want to take the chance of releasing a novel pathogen or a novel strain into the environment, so it was decided to treat the frogs with itraconazole prior to release. At this point, all the natural material was removed, and only plastic, plants, and materials that were easily disinfected were used. The yard was changed, and all of the frogs were consolidated to 5 of the stock tanks on one side of the yard, and they were treated and then moved to the opposite side of the yard while the original enclosure was disinfected. Initially, 1 stock tank was treated to make sure there would be no ill effects to the treatment, and these frogs did well, so all of the frogs that were housed at Woodland Park Zoo this year were treated. In the 5-gallon buckets, a disposable liner was used. 24 frogs were put in the bucket, and they were put in the itraconazole treatment for 10 minutes. The bucket was swirled to ensure that the entire frog was covered with the solution. And then they were released back to the fresh, clean, disinfected tub on the opposite side of the yard. And Virkon was used to disinfect everything, and this required 10 days of treatment, and the frogs did test negative at the end of the treatment. And this was a huge amount of labor that was used for this. I believe it was over 16 hours of labor per day, having at least 2 people in the yard every day to do this job with all of the disinfecting and everything. Some of the other health issues that we've had were golden tadpoles, golden eye, kinked tails and spine, extra limbs in metamorphic frogs, and some bloating issues. The golden tadpoles were much larger than the regular tadpoles, and they were unable to metamorph and died before actually turning into frogs. I don't believe that there has been any diagnosis or cause for this, but this has been seen in all of the partners that reared Oregon spotted frogs. Some of the frogs with severely kinked spines or extra limbs were either unable to metamorphose as well and were deemed unfit for release and humanely euthanized. And one of the issues that was seen at Northwest Trek and Mountain View Conservation Society in British Columbia, and British Columbia is not part of this working group, but I wanted to mention, since those 2 facilities had seen this similar instance of the bloating, the Mountain View Conservation Society submitted samples for analysis and determined that it was not a virus or bacterial infection, but pathology showed that the colon was not dispelling waste properly. And it happened at Northwest Trek and the Mountain View Conservation Society in the same year. And the assumption was that there was a cold snap soon after hatching that disrupted the development of the frog that allowed it to not dispel the waste properly in conjunction with genetic issues. Some of the other issues that we're seeing throughout the years are missing eyes or fused eyelids, partially undeveloped or extra limbs, which I mentioned with the kinked tails and spine, For the extra limbs, it's thought possibly to be a parasite, possibly from snails. Fused toe digits, air bubbles in the abdomen that often self-resolved within 24 hours, and a wilted, bent, or crooked tail that was sent for analysis and was a saprolegenia infection. So all these problems, of course, do not necessarily have answers, but thought to be genetic for some of them, environmental, and possibly husbandry issues as well. The year that we had the most of the kinked tails and spine at Woodland Park Zoo, it did seem that there was a genetic component since most of them were found from similar or from frogs within the same egg mass. So throughout the course of the project, we did continuous improvements every year. One of the things that we did at Woodland Park Zoo was look at the diet of the tadpoles. The original diet was a mixture of 50-50 kale with romaine lettuce mixed with spirulina. And we took that diet and fed that to half of the tadpoles, and the other half we fed these commercial Hikari algae wafers. And we found that the frogs that were fed the Hikari wafers grew in snout vent length faster than the tadpoles fed the original diet. So subsequent to this, we did continue to feed the original diet, but we supplemented each tank with the commercial diet as well. And as I said, over the course of the project, we did do continuous improvements for every step of the way. For example, the catch-up box that was used to catch frogs at the end of the season to bring to the wetlands for release was a simple shoebox lined with paper towels. And if you could imagine, you open the lid, put one frog in, that's great. For next frog, you catch up the frog, open the lid, put one frog in, the first frog jumps out. And then by the time you get to 3, 4, or 5 frogs, you have frogs jumping everywhere when you're trying to put one frog in. So a very simple solution Keith Knightman, one of the animal keepers that worked on the project, developed was just cut a hole in the top, have a simple deli cup fit in there while we're working that could easily be removed, the frog put in, all the frogs that are inside are still jumping everywhere, but you're not opening the entire lid. So the chances of the frogs jumping out of that hole were very low. And then there was that black piece of plastic that just fit over that hole that was rubber banded on for transport to the release site. So something very easy, very simple, very cheap that made our lives a lot easier. One of the other improvements that we made as tadpoles, initially the tadpoles when very small were housed in shoe boxes. These had very small amounts of water, which allowed for a much wider margin of, or a much smaller margin of error because there was not a lot of water to dilute any waste products or impurities in the water. It also increased the time needed to clean each of these shoe boxes for each of the facilities that had them. So this was changed to a floating net pen, and the tadpoles were released into these net pens, and there were 4 net pens placed in each of the stock tanks. So there was the full volume of water in the stock tank, and the tadpoles were housed in here. So there's a much greater volume of water that would dilute any impurities or any waste products in the water. And then we were able to use a turkey baster to manually remove any of the tadpole waste out of this net pen. So the tadpoles were reared in this pen until they were large enough to be safely released into the rest of the tank. And then back to this photo of the radiograph of the frog eating the other frog. To eliminate competition from frogs, we developed, again, very simple, very easy fix that had big impacts to the frogs. Just a simple deli cup with holes poked into it at different areas on it as a slow feeder. So insects are placed in here in addition to a broadcast feed. The larger frogs would be able to eat the frogs from the broadcast feed and be satiated much faster. The smaller frogs would then have a slow, steady release of food coming out of these holes and be able to have the nutrition that they need as well. And then some of the other equipment modifications that we made, again, simple, relatively easy, straightforward fixes. We increased the wattage of the heater so that the heater was not constantly overworking and burning out. So it improved the safety for the frogs of not having very wide temperature fluctuations if there were equipment failures. And then we had this mesh case that was made of stainless steel mesh that allowed the frogs not to come in contact with the heater for their safety. And then we also added these legs, which were simple stainless steel screws onto the bottom of the heater cage. And there were 4 legs on there, so it was steady on the bottom of the tank. Frogs would go under there, but the heater cage would not roll and crush any frogs. And we also had to improve water. I don't have a photo of this, unfortunately, but to improve the speed of doing water changes, a sump basket was created out of a similar stainless steel mesh, a little bit bigger than the mesh around the heater cage. And we used a sump pump to remove water, so it allowed for faster water changes, and it was safer that we didn't have to worry about sucking a frog out of the drain in the side of the stock tank. Even though we had a little filter outfitted over this, that it was much safer with the sump basket that was implemented. So over the years, survivorship at Woodland Park Zoo did steadily increase. Even with the chytrid fungus outbreak that we had, we still did steadily improve with all of the continuous improvements that we made to the program. And overall, 7,100 frogs have been released amongst all rearing institutions, all rearing partners. And again, all releases were done at Joint Lewis Base McChord Air Force Base. And they were released when large enough to avoid bullfrog predation, which is about 40 millimeters as per Washington Department of Fish and Wildlife guidelines. And as far as frogs being found in the area, they Washington Department of Fish and Wildlife does do surveys at this location, and they confirmed egg masses there in 2011, but none in 2012. No releases were done in 2013 as Fish and Wildlife was evaluating the program and just doing field studies for that year. And then for 2016 to 2019, no egg masses or adult frogs were seen at this area. However, in 2017, a study with Oregon spotted frog eDNA was performed, and there were 2 hits, 2 positive samples for the eDNA at the release site. So although they're not being found, there are still frogs that persist in the area. I don't know all the specifics of the eDNA, but I know it has a very small time that it would show a positive sign for. So it is pretty accurate in detecting species presence or absence. And that is everything that I have. So I would love to have any questions. And I would also just like to say thank you to a former Woodland Park Zoo curator, Jenny Pramick, who was part of the Oregon Spotted Frog program for its entirety, and all of her hard work and dedication that she put into the project. And she did share some of the photos in this project as well.